Under ideal conditions, the optode array provides high-quality data from all emitter-detector pairs throughout your experiment. In practice, this is not always the case. As a consequence, a data quality assessment is essential when working with high-density optical data. However, assessing data quality is challenging. There are many potential causes of data degradation, and it is difficult to define QA metrics that work well in all applications. NeuroDOT attacks this problem using a systematic approach. Briefly, the array data is presented for inspection. Then, a collection of QA metrics are calculated that identify anomalous source detector pair measurements. The effect of applying these metrics is summarized by plotting a subset of data in which flagged measurements have been omitted. This allows a visual comparison of the processed and unprocessed data. Finally, a qualitative summary is generated which superimposes QA results on the array and highlights potential problem optode locations. This allows the automated results to be reviewed, using knowledge of the data and goals of the experiment. The QA evaluation is an integral part of the NeuroDOT processing pipeline. Although a summary description is included as part of the data processing tutorial, a more thorough exploration is helpful to obtain a nuanced understanding of the QA process. For this reason, a separate tutorial has been prepared. Here we present the QA workflow in detail. We describe the types of plots provided, explain how the QA metrics are calculated, show examples of good and bad data, and outline the qualitative review. The goal is to identify an optimal selection of data to be included in reconstruction in order to obtain high-quality tomographic maps of neural activation. Before beginning, it's helpful to review two key concepts that will recur throughout the discussion. These are the concepts of array topology and source detector separation distance. The array optodes are distributed across the scalp in a three-dimensional configuration. However, several QA metrics depend more strongly on the two-dimensional relationship of optode pairs rather than their literal spatial position. This is conveniently summarized by considering a flattened layout of the array so that 2D relationships can be more easily visualized. For any source or any detector, we identify a nearest neighbor. The nearest source for a given detector, or in the example shown here, the nearest detector when considering a source. Because of the regular layout of the typical optical array, more than one detector usually exists at this minimum distance. Collectively, we refer to this set as the nearest neighbors. Using a similar construction, we can define the second nearest neighbors, third nearest neighbors, and so on. These categories ignore the three-dimensional positions of the optodes, emphasizing array topology rather than the array geometry. Nonetheless, the groupings provide a useful organization of measurement pairs in data quality assessment, as will be demonstrated later. A second key concept is source detector separation distance and its effect on optical measurements. This diagram depicts a light source and two detectors. Two features of the measurement provided by a detector depend critically on the distance to the source, referred to as RSD for source detector radius in QA figure labels and other documentation. Measurements provided by a more distal detector tend to exhibit decreased signal-to-noise because the light must traverse a longer path inside the tissue. Measurements provided by a proximal detector typically have better relative signal-to-noise ratio, but interrogation of the tissue may be poor because the light primarily remains close to the surface. Details may be found in the PowerPoint tutorial on image reconstruction. This trade-off is exploited by NeuroDOT to construct useful quantitative QA metrics as well as qualitative QA summaries, as will be demonstrated later in this tutorial. A good data quality assessment begins with a visual inspection of the data. The raw QA data plot generated by NeuroDOT is organized into two rows and two columns. The left and right columns show data at two wavelengths, 750 and 850 nanometers in the example used here. This is typical for optical imaging, in which data is acquired at multiple wavelengths, although the details will be system-dependent. Unprocessed data is plotted in the top row. The bottom row shows the results of applying QA metrics to exclude poor measurements. When working with task-based data, vertical green lines are superimposed which indicate stimulus onset. We will revisit the plots of good data after describing how the QA metrics are defined. The upper left panel is enlarged in the next slide in order to call attention to some notable features of these plots. The x-axis is time, or more accurately frame number, given the sampled nature of array data. The y-axis is light intensity, or more accurately fluence rate. Note the scale is logarithmic. The plot shows a selection of source detector pair measurements from the array. Because the traces tend to overlap, the content can be somewhat confusing. A handful of traces is shown expanded in the inset appearing here. Each trace represents one source detector pair measurement, which we will sometimes call a channel in what follows. The mean values of the time series typically span several orders of magnitude. Two examples are indicated on the figure. Note a smaller mean is generally accompanied by larger standard deviation. That is, lower amplitude measurements tend to be noisier. This effect is due to an increase in source detector separation distance, as discussed in the tutorial preliminaries. A default maximum separation distance of 40 millimeters is applied when selecting the channels that appear in the raw data plot. This provides a good overview of most data, although the parameter can be customized in the processing script. A proper maximum RSD value will be calculated based on QA metrics. These are presented in the next section. NeuroDOT uses a combination of time domain, frequency domain, and statistical analysis to identify poor quality data. The figure shown here summarizes the QA metric extraction. Some of these metrics comprise quantitative measures applied directly to exclude data, whereas others are more qualitative in nature and are used to construct a graphical synopsis for later review. Additionally, the QA code includes a number of parameters that tune the algorithm performance. Default values of these parameters are used in the tutorial and generally work well for a typical high-density optical array. However, these parameters may need to be adjusted when working with some types of data. As always, you may contact the NeuroDOT development team for help in customizing the software for your specific application. We begin with the plot at upper left, which displays a 10-second window of the largest amplitude measurements. A default amplitude threshold of 10 to the -2 is applied, and a maximum separation distance of 43 millimeters is considered when selecting these data. The rationale for this value will be explained later in this section. The expanded view permits closer scrutiny of the data, which should be examined for artifacts that may have been obscured when previously plotting data from the entire array. Often, small amplitude deflections are apparent in the expanded plot that repeat at approximately 1-second intervals. This signal is known to reflect the cardiac pulse and motivates the first of 3 metrics extracted from the data. An averaged frequency spectrum of the best quality data appears at lower left. The red and magenta traces show the average frequency content for short and medium source detector separation distance. The default values of 1 to 20 and 21 to 35 millimeters are used here. The average separation distance of these groups is 10.5 and 28, which are the values appearing in the legend. 2 spectral peaks are notable, which are particularly prominent in the medium distance measurements. The larger peak reflects regular presentation of the stimulus in this task-based example. This signal is generally not useful as a QA metric as it is strongly dependent on the experimental protocol. For example, it would not appear in resting state data. A second peak is centered at approximately 1 Hz. This frequency component is due to the cardiac pulse, which repeatedly distorts the optical signal via hemodynamic coupling, as was apparent in the expanded time series plot. Although it will be removed during processing, the cardiac pulse provides an excellent QA metric. Specifically, NeuroDOT quantifies the cardiac pulse using its band-limited SNR, that is, the summed power occupying a bandwidth from 1/2 to 2 Hz divided by the power in the 2 immediately adjacent bands. Data from the longer of the 2 applied light wavelengths is used, as this generally exhibits superior signal-to-noise ratio compared to shorter wavelength data. Note the frequency cutoffs can be customized in the processing script. Although the cardiac pulse usually provides good results, other frequency content is sometimes used, such as the functional connectivity band when working with resting state data. See the PowerPoint tutorials for details. The second metric used in the QA evaluation is derived from an analysis of light falloff as a function of increasing source detector separation distance. Theoretically, the light intensity from a point source decreases exponentially with distance. If we assume the optical properties of the medium are isotropic, multiple measurements acquired at the same separation distance should be identical. Such measurements are available in array data because of the regular spacing of the optodes. For example, all second neighbor measurements should be equal, because all are the same distance from the source. This would be true for all second neighbor measurements combined from all sources in the array. However, the medium in a neuroimaging experiment is not isotropic, and so any radial symmetry is only approximate. As a result, multiple fluence rate measurements at a common separation distance will spread around a mean value. This effect is exploited to define a second key QA metric summarized as a fall-off plot. An example is shown here at upper right. In this figure, source detector separation distance is plotted on the x-axis and increases left to right. The y-axis is logarithmic. Each point is the mean light level across all frames for one measurement, and the color indicates the light wavelength used in data collection. Note the points do not spread continuously in the x direction, but rather cluster at discrete separation distances, representing nearest neighbors, second neighbors, and so on. As should be expected, the mean of each group decreases with increasing separation distance. However, beyond some maximum value, no further decrease is observed. The effect is due to a noise floor. In this regime, a given detector no longer responds strongly to its paired source. Rather, the signal is dominated by the optical background and other noise common to multiple array channels, a phenomenon referred to as crosstalk in the NeuroDOT documentation. The transition to the noise floor defines a second useful metric applied in QA evaluation. For the data shown here, we estimate a maximum useful source detector separation distance of 43 millimeters. This value is applied elsewhere in the QA evaluation. For example, it was used when selecting data to display in the expanded time series plot described earlier. It will also be used in later analysis. A second QA measure supplied by the fall-off plot is based on the spread of a given group about its mean value. Specifically, the spread should generally be no more than 2 orders of magnitude. This is not applied as a quantitative metric, but rather has been established as good practice based on past experience. It should be considered when visually reviewing the fall-off plot constructed from your data. The final QA metric generated by NeuroDOT is based on channel noise, as quantified by the standard deviation of a source detector pair. Recall that optical data tends to exhibit a range of signal and noise, as is readily apparent in a plot of the raw data shown earlier. A decrease in mean channel value is generally accompanied by an increase in standard deviation. A small standard deviation indicates good data quality for the simple reason that physiological optical signals generally vary by no more than a few percent. Conversely, a relatively large standard deviation suggests the measurement is dominated by noise and likely should be excluded from processing. To quantify this distinction, a histogram of standard deviations of the array data is constructed, and channels having values exceeding a predefined threshold are flagged. The x-axis is the standard deviation of the measurement following normalization by applying the Rythoff approximation, and the axis is scaled so that the values may be interpreted as a percent. A 7.5% threshold has been found to give good results, and so is used as the default cutoff. However, the value can be changed in the data processing script. Good data will have most or all measurements to the left of the red line in the histogram plot. In summary, the QA plot provides opportunity to inspect the time series data, its spectral content, the array light level fall-off, and the channel noise quantified by standard deviation. 3 quantitative metrics have been constructed: the average signal-to-noise ratio, the maximum recommended source detector separation distance, and a list of channels exceeding a recommended standard deviation threshold. The example used here is typical of good quality array data. For comparison, an example of problematic data is shown in the next slide. Note several of the traces in the time series plot exhibit notable drift. The cardiac pulse is not apparent in the spectrum. There is large spread of mean light levels in the fall-off plot, and the standard deviation of many channels exceed threshold. As a consequence, many of these measurements will be excluded from processing, which may result in a poor quality reconstruction. However, to better understand the results of the QA evaluation, we need to consider how the metrics are applied. First, single-channel data is graded based on standard deviation. Specifically, a given channel will be included in processing if its standard deviation is less than threshold, which is generally fixed at 7.5% as shown earlier. Signal-to-noise and max separation distance are not part of the single-channel classification, although the metrics will be used later in the final QA review. The effects of applying this criterion can be reviewed in the raw data figure shown earlier. The bottom panels exclude measurements that failed the single-channel classification. A comparison of the top and bottom rows shows that channels exhibiting a large standard deviation have been removed and that the time series means are larger overall. Note the difference in the y-axis scaling. The results shown here are typical of the improvements that are obtained. Good data is characterized by traces that exhibit a relatively constant standard deviation across all frames. Obvious inflections or variations, if present, should coincide with the stimulus event when working with task-based data. Conversely, data that drifts over time or changes variance or contains artifacts, especially artifacts that span multiple channels, indicates poor optode coupling or other quality issues. Remediation of such data is sometimes possible as part of statistical analysis or through custom processing. For example, offending frames can be identified using a motion metric and deleted prior to processing. On a final note, the example shown here is particularly problematic in that NeuroDOT did not flag these measurements. Although the QA metrics used by NeuroDOT are generally reliable, some types of artifacts may evade detection, given the variety of potential noise sources and their sometimes unpredictable effects. Such results highlight the importance of visually reviewing your data at all stages of the QA evaluation. The NeuroDOT Data Quality Assessment has identified source detector measurements that will be excluded from further processing based on applying QA metrics extracted from the data. A graphical summary of the results, such as the example shown here, is generated for review. At top, two key QA measures are superimposed on the array. At bottom, a graph of the array is displayed, with the optodes drawn as nodes and good measurements drawn as edges. The layout of the summary figure is based on the flattened array topology and reflects the optode configuration used in data collection. Some examples featuring more complex arrays are shown here. In this final section of the tutorial, we explain how this information can be used to evaluate the QA analysis, including how it applies to the current data and how it might be used to improve data acquisition in future experiments. The mosaics shown in the top half of the figure summarize mean light level and SNR, calculated at each optode position and plotted using a pseudo-color tiled map. Each tile value is the light level or the signal-to-noise ratio at a given optode position, averaged across all measurements that include the optode, whether the optode is a source or a detector. All applied wavelengths are used in calculating the mean light levels, whereas only the longer wavelength is used in calculating SNR. Additionally, signal-to-noise is based on the bandwidth used earlier to define the SNR metric. The default value used by NeuroDOT isolates the frequency content of the cardiac pulse. The left and right columns plot results restricted to short distance and medium distance source detector separation. The default values selected for these classifications are 1 to 20 and 21 to 35 millimeters. These have been found to give meaningful information in most applications. The color scaling is defined so that brighter colors represent better parameter values. The plot at bottom is a graph of the array with optodes as the nodes and each good measurement drawn as an edge connecting the participating source and detector. The primary feature of interest are the missing edges. An edge is not drawn if the data for that source detector pair failed the QA criteria applied in the evaluation of individual channel data. The distribution of good and bad channels is then used to identify bad optodes. Specifically, an optode is flagged as bad if more than 1/3 of the measurements it participates in were flagged as bad. That is, 1/3 of all detectors if the optode is a source, or 1/3 of all sources if the optode is a detector. The threshold can be changed in the processing script to be less or more stringent, but 1/3 has been found to give good results in practice. Optodes classified as bad according to this criteria are circled. Note that two separation distances appear in this summary. The percent good measurement calculation uses the maximum separation distance estimated in the fall-off analysis. However, a smaller separation distance is used in optode classification because it generally depends on the percent cutoff parameter. The default values of 33% and 33 millimeters have been found to work well together and give good results for typical array data. We now consider how the information presented in this figure is used. As a rule, the light level and SNR plots are sometimes redundant and sometimes complementary. Good data typically results in 4 uniformly bright plots and no further action is necessary. Only when the data includes at least a few poor measurements are subtle and sometimes not so subtle effects notable. First, we consider the difference between the short and medium range measurements. We can interpret short range measurements as physics, whereas medium range measurements reflect a combination of physics and biology. This is an oversimplification, but it is grounded in how light propagates inside the skull. Light traveling to a detector close to a source remains close to the surface. As a consequence, information about cortical activity is primarily carried by the medium distance measurements. As a rule, good short distance measurements are a prerequisite to obtaining good medium distance measurements. This is simply because there are more factors that affect the latter. However, good short-distance measurements are necessary but not sufficient. For example, if the scalp contact of a given source optode is sufficiently poor to compromise short-distance measurements, it will also compromise medium-distance measurements because light level decreases and noise increases with increasing source detector separation. For this same reason, it's possible marginal source coupling may compromise medium-distance measurements while sparing short-distance measurements. A few areas of the array demonstrating this phenomenon have been highlighted in the example shown here. Bright tiles can be identified in the short-range measurements with a corresponding dark tile, that is poor data, in the medium-range plot. The effect is generally more conspicuous and more consistent in the SNR data. In fact, a contradictory pattern is observed here in some parts of the mean light-level mosaic. This is more likely to occur at an array edge where a single marginal optode can more strongly affect the result, because it is based on fewer measurements. The mean light level and SNR mosaics tend to be more consistent in the medium range measurements, with a problem area in one typically corresponding to a problem area in the other. Correspondence in the short range measurements is generally less predictable. In fact, the short range SNR is typically the most forgiving metric. As noted earlier, it is a necessary but not sufficient indicator of all of the other metrics. Finally, regions of poor light level or poor SNR tend to coincide with the locations of poor optodes identified in the array graph. This should not be surprising, as both quantities contribute to the evaluation of individual source detector measurements, which in turn are used to identify poor optodes. As such, the array graph is to some extent redundant. However, the thresholding applied to identify poor optodes provides a handy summary, condensing complex, continuously varying QA measures into a discrete map highlighting problem areas that can be used to guide data remediation. The primary goal of the data quality assessment is to exclude poor measurements from further processing so that only high-quality data contribute to reconstruction and tomographic mapping. But a second goal is remediation. That is, how the QA results may be used to improve the data. The response divides broadly into improving the current data or considering steps that may be taken in future experiments. If QA has flagged some of the array optodes, the ideal response is to fix the problem. Many issues are the result of poor array fit, which can be improved using the QA summary figure to identify areas of poor array coverage. This requires the data quality analysis be carried out during the recording session, but such an expectation is not entirely unrealistic. The NeuroDOT QA code executes quickly, so results could be made available within the time frame of the typical imaging experiment. Additionally, many contemporary acquisition systems include a live display of optode coupling or other metrics that provide immediate feedback to help avoid data quality issues. Still, we recognize this solution is not possible in many situations. It is not an option if analyzing the data after acquisition is complete or when working with public data downloaded from a website such as OpenNeuro. A second remediation option is to work around the problem. This is sometimes possible if the region of poor array coverage flagged in the QA analysis lies outside the area of interest defined by the goals of the experiment. An example is shown here, which depicts the QA summary for a two-part array featuring a separate pad for recording from the motor area. Poor coverage is largely confined to the motor pad. In this case, remediation involved simply omitting the motor area from the reconstruction and subsequent analysis. This solution may not be ideal, but occasionally, a compromise must be made in order to maximize the data available for study. A final remediation option is to consider an alternate analysis. The rationale for high-density diffuse optical imaging and the goal of NeuroDOT is the generation of tomographic maps of brain activation. However, tomographic mapping is the most demanding application of optical data. If the QA analysis has resulted in substantial data loss, there may be no option but to consider other uses of the data. This includes low-resolution tomography, 2D topography, or even basic time series analysis using what few isolated source detector measurements have survived the QA assessment. The choice depends on both the quantity and quality of the available data, as well as the goals of the experiment. In the worst case, it may be that the data is simply unusable. You always have the option of rerunning the data quality evaluation after adjusting script parameters to apply more lenient thresholding. However, the default values, although largely derived empirically, have been found to give good results in prior use of NeuroDOT. In this case, a better option may be to take lessons learned from poor results to inform the planning of future experiments and avoid additional problems as the study proceeds. For example, if the QA analysis consistently flags the same optodes or the same array region across multiple subjects, it may indicate faulty hardware or a systematic error in array placement. These issues should be investigated and addressed before data collection proceeds. Parenthetically, a varied distribution of flagged optodes is preferable, as not only does this argue against a hardware issue or experimenter error, but it is less likely poor coverage in the individual subject maps will be an issue once they are combined in a group analysis. Lastly, poor QA results can draw attention to individuals in your subject pool that may continue to present challenges in future experiments. For example, children and older adults generally exhibit more movement during recording than young adults, so you might plan for additional scrutiny when working with this subject pool. Finally, hair is a perennial problem in optical imaging. Some subjects present more of a challenge than others to obtain good optode contact with the scalp. These subjects will require more preparation time and care, whether an initial visit or returning in a longitudinal study. In summary, the primary goal of a data quality assessment is to identify which array measurements to include in processing and reconstruction. The QA metrics computed by NeuroDOT are generally reliable, but they are not a substitute for visually inspecting your data. Automated evaluation consists of calculating quantitative metrics from the data, which are applied to identify poor channels and problematic optodes, which in turn identifies areas of poor array coverage. These results are presented for review to help the user make informed decisions about the current data and potentially improve future acquisition. The data quality assessment is an integral part of the data processing pipeline. As a next step, you may wish to review the 2 data processing tutorials, starting with an overview that summarizes how the different tutorials fit into the overall picture, followed by the full data processing tutorial that presents the processing pipeline in detail. Finally, when you want to begin hands-on training or simply want to explore details of how QA is coded in NeuroDOT, the PowerPoint tutorial listed here offers self-guided instruction using an accompanying MATLAB script which implements QA, pre-processing, image reconstruction, and visualization, working with sample array data included with the distribution. Additional information may be found in the resources listed here. Feel free to contact the development team with questions or post them in the comment section below. Thank you for your interest in NeuroDOT.